Hello everyone, welcome to this tutorial. Today we're gonna learn sequence complement and reverse complement, how to do transcription and translation on DNA sequences. So the first thing we do, like we did before in sequence parsing, we here import the bio.seq. From bio.seq import seq. We run this cell. First, for example, if we have this sequence, this is a DNA sequence, and we need to get the complement of this sequence, which means the complementary nucleotides on the other strand, on the other DNA strand. So first we define our sequence. It is in this variable myseq. Then we just run this method, which is myseq.complement. We run, and here we get the complement of our sequence. This is the complementary nucleotide sequence of this sequence. So what if we want to get the reverse complement? We just run this method, myseq.reverse complement. We run, then we get the reverse complement. Is just reversing of this complement sequence here. As we can see here, GCG, and here this GCG is on the left side, CTA on the right side. Here CTA is here on the left side, and here is on the right side. And this is the reverse complement of the original sequence myseq. Next, we will see how to do the transcription of our sequence. Transcription is to convert the DNA sequence into RNA sequence. So we have here the coding DNA, starting with ATG. This is the start codon, the standard start codon, and TAG, this is also a stop codon. Suppose this is a gene sequence, this is a coding sequence. So we use this coding DNA sequence. We want to do a transcription. So first we define our coding sequence, and next if we want to get the reverse complement like we have done before, so we just run this cell. It will give us the reverse complement and store it in this variable template DNA. Now we want to get the transcription of our sequence. First, we want to copy this, and we need to look at the transcription of our sequence. First, we had run this code. Now we want to get the transcription of our sequence. Run this, and here we have the RNA sequence, or the transcription of the DNA sequence. As we can see here, it's just converted the thiamine into uracil. That's all what happens. In translation, all T is converted into U. So what if we want to return it back or if we have an RNA sequence and we want to convert it into a DNA sequence, this is very simple in Biopython, is done through this method, back transcribe. So the first method which we use, the coding DNA dot transcribe. This method transcribe gives us the RNA sequence, and back transcribe returns us back to the original sequence. As we can see here, this is the original coding DNA sequence which we started with. So we can do transcription and reverse transcription using transcribe and back transcribe methods. So next we need to do translation, and translation, as we know, means conversion of the RNA sequence or the DNA sequence into protein sequence. In Biopython we can do this process both on DNA sequence and RNA sequence. In nature it happens only for the RNA, not direct translation to the DNA, but in Biopython we can do it directly on DNA or RNA. As we can see here, we have a sequence of mRNA or messenger RNA, a messenger RNA. We just define it at the beginning, and then we use the method which is messenger RNA dot translate. Use the method translate. So the method is just intuitive: transcribe, back transcribe, and translate. The names are just intuitive. We can easily recall the name of these methods. So dot translate, we get here the translation of this RNA sequence. The translation here, we notice that we have two asterisks in this sequence. The asterisk in the translation means stop codon, and if we look here in the original DNA sequence, we will find the an actual stop codon here, this one, in the middle of the sequence, TGA, and another stop codon at the end of the sequence, TAG. So here, as we can see here, Biopython identifies the stop codons and inserts asterisk inside the sequence or at the end of the sequence if it finds a stop codon like TGA or TAG. So here this is the translation, the protein sequence which came out from this DNA sequence or RNA sequence. Also, as I said, we can do it directly on the coding DNA, so we can run the method translate directly on the DNA sequence, and we get the same result as we got from the messenger RNA dot translate. So it can be run on both DNA or RNA sequence. So this method translate, it has some arguments. So if we press Shift Tab, we get the docstring of this method or the signature of the method. We see here the dot translate method have a number of arguments, like the first one is the table, second one is the stop symbol, third one is the to stop, this is a boolean, either true or false, and CDS, also this is a boolean, either true or false, and gap. So this is a number of arguments we have to define if we want to get some specific results out of our translation. For example, here we define the table. The table is means that the table of the genetic code. We know that the genetic code differs among different organisms, and there are different genetic code tables. We can see them here on this website. The genetic codes is present on the NCBI website, and we can see here they have more than they have 33 tables of different genetic codes. As we can see here, this is the standard one, and the vertebrate mitochondrial code, the yeast mitochondrial code, and so on. So here we have this is there are different tables for different translations of genetic codes among different organisms. So if we want to translate our DNA or RNA sequence according to different or specific table among these tables, we also can specify this in the translate method. So we define the table. Either we use a string like this is a vertebrate mitochondrial, or we use according here we see this, the vertebrate mitochondrial table here is table 2, so we can change this into 2, and we will see now. So we can see coding the translate table vertebrate mitochondrial, run, we get this method. As we can see also, this is the same, this is the coding DNA, this is the same DNA sequence, but here it did not identify TAG here, did not identify TGA here as a stop codon, because this is a different table for translation. But in the when we use here translate without any arguments, it used the standard table as default. As we can see here, when we press shift tab, we get here the table is defined as standard. This is the default for this method, and in this case the default table defines the TGA as stop codon. So this is in the middle of the gene, there is a stop codon here. According to the vertebrate mitochondrial table, TGA is not defined as a stop codon, so we can see here it only defined TAG as a stop codon. So as we can see, it depends on our table. The outcome of the translation depends on the table which we use. Also, we can replace this by number 2. For example, this is the same table here. The vertebrate mitochondrial table is table number 2. So we can type only 2 and run. So as we can see here, run, we get the same result. There's no any difference because this is the same table. So we can either use 2 or vertebrate mitochondrial, and we can get it here. This is the code here. We just add the integer, the number. We don't use this one. We just use the integer. So there is another argument which is toStop. toStop, this is a Boolean argument which is either true or false. As we noted here, we have 2 stop codons in the middle, one in the middle and one at the end of the sequence. So in nature, DNA is translated until it reaches the first stop codon, and then it will stop here. It will not continue until it finds the next stop codon. So this is what happens in nature. So if we want to do the same thing like what happens in nature, we translate our sequence to until it reaches the first stop codon. So we define this argument as true. to stop. So we will run this method and see here. As we can see here, we get the sequence until it reaches the first stop codon. Here this is arginine amino acid, and here followed by the stop codon, and as we can see here, it stopped here. So to stop, in nature actually the stop codon is not included in the DNA sequence, in the protein sequence. So here it does the same. When it reaches the stop codon, it will stop here. There is another thing, which is suppose we have a bacterial DNA sequence, and in some bacteria the start codon is not like the start codon which we are used to, that ATG. This is the start codon in the standard table, and as is commonly known that ATG is the start codon which is coding for methionine amino acid, as we can see here. So in some bacteria like E. coli, the standard start codon could be a different one, like this one, GTG. Actually this is a start codon, and in nature it codes for methionine, and the same thing. Actually, as we can see here, if we run this, we have run this DNA sequence. Now we will see what will be the outcome of this. If we want to translate this, we should select table as bacterial and then run. Then we get the sequence in this way. What happened here? We see that here the GTG coded for valine amino acid. Normally GTG codes for valine in bacteria, as if it found here inside, as we see here in the middle of the protein sequence, there are some valine amino acids, valine residues present here because of the this codon GTG. But at the beginning, this should be coding for methionine, not valine. So if we run this, we select bacterial, and to stop, that is true. So actually here it doesn't include the asterisk because we define to stop is true. But also we still have valine as the first amino acid, and we will see how to change this into methionine. Here this can be changed by using the CDS argument, as we see here in the method docstring. This is the argument here. CDS is a Boolean. CDS means I define this sequence as a coding sequence, which means that Biopython will be searching in this sequence for the start codon, the stop codon. As we can see here, it's TEA. This is a valid stop codon, as we know. But this start codon is not standard start codon. This start codon is not standard. It's only in some bacterial species like E. coli. So what we want to do here, we will tell Biopython that this is a coding sequence. So Biopython is going to search for this and search for the bacterial table for its start and stop codons, and if it finds this GTG as a start codon in bacteria, so it will give us the valine, it will give us methionine instead of valine, because this is a valid start codon in bacteria. So first, if we want to get this result, we have to define the table first, this is bacterial table, and second we have to define that this is a coding sequence. So Biopython should search for the beginning of the sequence as start codon if it is a standard or not in the defined table, and at the end of the sequence it will search for the stop codon, and also it will search the length of the sequence if it is a multiple of 3, because it must be a multiple of 3, because each 3 nucleotides will be translated into only one amino acid. So we run this, we get the protein sequence. As we can see here, valine is changed into methionine because we have defined that this is a coding sequence and this is a bacterial gene, which means that GTG is a valid start codon, and therefore we got methionine instead of valine. In this case, if we don't define that this is a coding sequence, it will not give us this result. It will translate GTG into valine, not methionine. Thank you, and see you in next videos.